If you've ever opened a research paper and thought, I want to understand this, but this is dense, then this podcast is for you. Every day, thousands of research papers are published. Most of them are rigorous, insightful, and almost impossible for non-specialists to read comfortably.
Oh, absolutely. It can be a lot.
But the ideas inside them shape medicine, technology, business, and society. Welcome to One Paper Orbit. We are a research paper podcast. Here, we take peer-reviewed research and turn it into clear, structured conversations without adding speculation, without exaggeration, simply making the original work easier to understand.
Just sticking to the facts,
Today, we are discussing a paper authored by H.K. Chung, Kong Liu, and a large collaborative team of researchers titled Atlas-Guided Discovery of Transcription Factors for T-Cell Programming, published on February 2026 in Nature.
It's a really incredible piece of work.
This discussion is based on a paper published under the Creative Commons Attribution 4.0 license. The original authors retain credit for their work. This podcast provides an adapted explanation for educational purposes, with full citation and license information provided in the description.
Perfect. So let's get into it.
Welcome to the deep dive. So, have you ever wondered why your own immune system, why it can't always just fight off things like cancer naturally? I mean, you have this incredibly sophisticated defense mechanism built right into your body.
Right, millions of years of evolution.
Exactly. Yet sometimes it seems to stop working entirely. And today, we are looking at a paper that attempts to answer that exact question. We are exploring how scientists are mapping the molecular master switches of our immune system
To basically engineer better, stronger cancer-fighting cells. It's a fundamental question in immunology, really. Because to understand how scientists are engineering these better therapies, we first have to understand the cellular workforce.
The ones actually inside your body doing the heavy lifting.
Yeah, doing the fighting. And more importantly, we need to understand why some of those workers mysteriously just, burn out on the job. And the primary workers we're talking about today are a type of white blood cell called the CD8 T cell.
Okay, let's unpack this. If we think of your immune system as this massive factory, these CD8 T cells are kind of like the heavy machinery operators, right? They're the ones physically out there seeking and destroying infected cells or cancer cells.
That's a great way to picture it, yeah.
But they don't just do the exact same job all the time. They adapt based on what they are fighting.
They are highly, highly adaptable. When a T cell encounters a threat, it differentiates. It actually changes its physical state. So, like if you catch a quick acute infection, say a seasonal flu, your T cells ramp up, clear the virus, and then form a memory of it.
So you don't get sick from that exact strain again.
Exactly. But a solid tumor is a completely different environment. It's not quick. It's a chronic long-term threat. The T cells are constantly being stimulated, constantly being told to fight just day after day after day.
Right. And this paper focuses heavily on two very specific states that these T cells get pushed into when they are stuck inside that kind of chronic tumor environment.
Yes, exactly. So the first state is called the tissue-resident memory T cell. The paper refers to it as the TRM cell. You can think of these as your highly productive elite workers.
The ones you really want on the factory floor.
Oh, absolutely. When oncologists look at patients with solid tumors, having a high number of these TRM cells inside the actual tumor tissue is strongly linked to significantly better survival rates. They dig right into the tissue, they produce the right chemicals, and they fight the cancer effectively.
Okay, so if you have cancer, you obviously want your factory packed with these elite TRM workers. But there is a second state, right? Which is essentially the dark side of this whole process.
Yeah, that is the terminally exhausted T cell, or TEXterm cell. In an environment with a persistent, overwhelming threat like cancer, the T cells are just totally overworked. Eventually, they hit this biological wall and they burn out.
Wow, so they just stop working entirely.
They become completely dysfunctional, they stop dividing, they lose their ability to attack, and they start expressing these inhibitory receptors on their surface proteins like PD1 and TIM3.
Wait, hold on. Why would our bodies even have a system that allows our absolute best immune cells to just become exhausted and give up? That seems like a massive biological flaw.
It sounds like it, right? But it actually evolved as a really crucial safety mechanism. If your immune system just stayed turned up to its maximum lethal capacity indefinitely, you would literally start destroying your own healthy organs.
Oh, wow. So it's protecting you from yourself?
Exactly. Exhaustion is the body's way of putting the brakes on to prevent extreme autoimmune damage during a really bad chronic infection. It's a feature, not a bug. The problem, though, is that cancer has evolved to exploit this safety brake. It basically forces the T cells into this exhausted state so the tumor can survive.
And once they are in this exhausted state, these TEXterm cells fail to respond to our best current immunotherapies. They just sit there on the factory floor, completely useless. So, logically, to cure the cancer, we just need to find the specific genes that cause the cell to become exhausted and turn them off.
That is the ultimate goal, sure. But that brings us to the central paradox of this paper. What's fascinating here is that these two cells, the elite productive TRM cell and the completely useless exhausted TEXterm cell, are remarkably similar.
Wait, similar how?
Well, they both prefer to live deep in the tissues, for one. And when researchers looked at their key transcription factors, which are, the proteins acting as the master switches to turn specific genes on or off, they found that both the elite workers and the burned-out workers shared the exact same major switches.
Hold on, I don't buy that. If one cell is actively saving your life and the other is basically a couch potato actively hindering your immune response, there has to be a massive physical difference in their DNA blueprints. Are you saying they are literally identical?
I mean, they share an astonishing amount of their regulatory blueprint. They both rely heavily on transcription factors with names like BLIMP1, BHLHE40, and NR4A2.
That's a mouthful.
Yeah, immunologists love their acronyms. But the similarity goes even deeper than just those switches. It goes down to the physical structure of the DNA itself, which is a property called chromatin accessibility.
Okay, let's explain chromatin accessibility for the listener, because that sounds incredibly dense. What does that actually mean in plain English?
Sure. Think of your DNA like a massive library of instruction manuals. If a book is closed and locked inside a vault, the cell's machinery cannot read it. That is closed chromatin.
Right, makes sense.
But if the book is open and sitting on a desk, the cell can read those instructions and build whatever protein is on that page. That is open chromatin. What this paper confirms is that the elite TRM cell and the exhausted cell have highly correlated open chromatin regions.
So they essentially have the exact same library books open on their desks.
Exactly.
But if they look that identical, how is it physically possible to tell them apart, let alone treat the patient? Like, if you try to invent a drug that targets one of those shared master switches to stop the exhaustion, wouldn't you accidentally just wipe out the protective cells too?
Yes. And that is exactly the wall the previous researchers hit. Because the cells are so similar on a broad level, earlier studies missed the incredibly nuanced differences that actually separate their fates.
Oh, I see.
The researchers behind this paper realized they couldn't just look at the basic high-level blueprints anymore. They needed to build a comprehensive, multi-layered map of the T cells' inner workings. They had to build an atlas.
Okay, but an atlas of what? If the DNA is the same and the open library books are the exact same, what are we actually mapping here?
They mapped the actual influence and activity of the genes across 121 different samples, representing 9 different states of CD8 plus T cells.
Wow, that's a lot of data.
It's massive. And to build this multi-omics atlas, they combined 2 very powerful sequencing techniques. The first is RNA-seq, which measures gene expression.
Okay, so RNA-seq tells us which genes are actively being copied and translated into proteins. If we go back to our factory analogy, it's like measuring how loudly a particular manager is shouting orders on the floor.
That's a great way to look at it, yeah.
Yeah.
But shouting isn't enough to understand true influence. A transcription factor might be produced in huge quantities, shouting really loudly, but it might lack the necessary cofactors.
Like helper molecules it needs to function.
Right. Or the specific DNA target it is trying to bind to might be locked away in closed chromatin, so nobody can hear the shouting anyway.
Which is why they also use the second technique, the ATAC-seq.
Yes. ATAC-seq maps out the chromatin accessibility we just talked about. It shows exactly which parts of the DNA are physically open.
Got it.
By combining RNA-seq and ATAC-seq, the researchers weren't just looking at who was shouting the loudest. They were looking at who was actually standing in a room with open doors, able to be heard by the rest of the cell.
But taking all of that raw data, the shouting managers and the open doors across 121 different samples, sounds like an incomprehensible amount of information. How do you actually find a meaningful pattern in all of that noise?
You have to use computational biology. They fed all this data into a bioinformatics pipeline called TIG, and TIG constructs what is known as a gene regulatory network.
Okay, what does that actually look like?
Well, instead of just looking at individual genes in isolation, TIG models the complex social network of the cell. It maps out how a transcription factor connects to its target genes and how those genes connect to others.
Wait, PageRank? Like the algorithm Google uses to rank web pages, so we aren't just looking at how loud a gene is shouting, but how many other important genes are actually linking to it and listening.
That is the exact underlying mathematical principle TaiG uses. It applies a personalized PageRank algorithm to the genetic network.
That is fascinating. So, like, in the early days of the internet, a search engine might rank a web page highly just because it repeated a keyword 1,000 times. That's kind of like RNA-seq, just measuring how loud a gene is shouting.
Right. The early internet was easily tricked.
Yeah. But then Google realized a web page is truly important if other highly important web pages are constantly linking to it.
And the pipeline applies that exact same logic to biology. It assigns an important score to every single transcription factor by calculating its global influence within the entire regulatory landscape.
That's brilliant.
It really is. It looks at how many downstream targets a factor controls and how many upstream regulators are feeding into it. It finds the true master switches driving cell differentiation, even if they aren't the most highly expressed genes in the cell.
They essentially created a metric for biological clout. So once they ran this massive PageRank analysis on the T cell's social network, what did this map actually reveal? Did they find the differences between our identical twins?
They did. The statistical filtering categorized the transcription factors into two main buckets. First, they found 173 multi-state transcription factors.
A multi-state, meaning they're everywhere.
Exactly. These are broadly active across many different cell types. Going back to your factory analogy, these are the general plant managers. They just keep the lights on and the basic machinery running.
And I imagine these are the ones previous studies kept stumbling over because they're so loud and everywhere.
Exactly. They were masking the real drivers. The true breakthroughs were the 136 single-state transcription factors they discovered.
Ah, so these are the hyper-specific ones.
Yes. They were the highly specialized shift supervisors that only show up under very specific conditions. Each of the 9 T-cell states they studied was driven by its own unique set of these single-state switches.
Here's where it gets really interesting. How did the map break down for our elite protective cell and our burnt-out exhausted cell?
Well, the pipeline isolated 30 transcription factors that were shared between them, which again highlights why they look so similar on the surface. But crucially, it identified 20 switches that were active only in the protective TRM cells.
And the exhausted ones.
It found 34 switches exclusive to the exhausted TEXterm cells.
Okay, let's name drop a few of these newly discovered shift supervisors. For the protective cells, I know the pipeline flagged a unique regulator called KLF6. For the shared regulators, they found HIC1 and GFI1. But what about the exhausted cells? What is actually causing them to give up?
The pipeline identified 2 entirely unprecedented drivers of the exhausted state, ZSCAN20 and JDP2.
Wait, unprecedented? Like nobody knew what they did before this?
Correct. Prior to this paper, these 2 transcription factors had absolutely no known function in T cells. They were flying completely under the radar.
So ZSCAN20 and JDP2 are the corrupt shift supervisors. When they get activated by the chronic stress of the tumor, what are they actually doing to the cell to make it exhausted?
They physically bind to the DNA, and they alter the cell's programming. They force the cell to upregulate those inhibitory receptors, the molecular white flags like PD-1 that we talked about earlier.
So they're throwing up the white flag.
Yeah, and they simultaneously shut down the genes responsible for producing the chemical weapons the cell actually needs to kill the cancer. They actively command the factory workers to stop fighting.
But because these 2 bad managers are only present in the exhausted state, does that mean researchers finally found the exact individuals to fire? Like, by computationally isolating ZSCAN20 and JDP2, did the researchers essentially find the exact wires to defuse the exhaustion bomb without turning off the immune system's security cameras?
That is the theoretical promise of finding single-state transcription factors. Yes, it offers precision. You can alter the disease state without collateral damage to the healthy protective states.
It promises it on a computer screen, sure. But we all know a computational prediction is just a hypothesis. The map is not the territory. Predicting a master switch on a computer is a great math trick. But does this actually cure a living, breathing subject?
That's the billion-dollar question. The researchers knew they had to prove the map worked in reality. So they moved from the digital realm to living organisms. They set up an incredibly rigorous test using a technique called in vivo Perturb-seq.
Okay, Perturb-seq involves using CRISPR, the gene editing tool, right? How does that work in this context? We aren't just cutting random wires.
Right. They took healthy T cells and used CRISPR to edit their genetic code, physically deleting the instruction manuals for these specific transcription factors.
So making custom batches of cells.
Exactly. They created different batches. Some T cells were engineered to lack the shared switch HIC1, some lacked the new exhaustion switch ZSCAN20, and others lacked JDP2.
They literally fired the specific managers before putting the workers on the floor.
Yes, exactly. Then they transferred these edited T cells into live mice that were battling B16 melanoma, which is a very aggressive form of solid cancer.
Yeah.
They let the T cells fight the tumor and then track the outcomes.
Did deleting the instructions for ZSCAN20 and JDP2 actually stop the T cells from becoming exhausted in the mice?
The results were highly definitive. When they deleted these specific exhaustion switches, the T cells exhibited significantly enhanced tumor control.
Oh, wow.
Yeah. The tumors in those mice were demonstrably smaller, and the overall tumor weight was reduced. By removing just those corrupt shift supervisors, the T cells were able to fight longer and harder in the chronic tumor environment without burning out.
That is incredible. But we have to address the other half of the paradox. What happened to our elite protective TRM cells? Did deleting those exhaustion genes accidentally ruin the good cells?
No, it didn't. And that is perhaps the most crucial validation of the entire TAICHI pipeline. Deleting ZSCAN20 and JDP2 suppressed the exhausted state without interfering with the formation or function of the protective tissue-resident memory cells.
Wow, so they completely separated them.
Yeah, the researchers successfully decoupled the two programs.
So the precision actually held up in a living animal. But the paper also mentions this genetic rewiring synergized with existing therapies. I know we talked about checkpoint blockade drugs earlier. Can we explain how those actually work and how this new discovery ties into them?
Certainly. As we mentioned, tumors often wear molecular fake ID badges, proteins like PD-L1. When these fake IDs bind to the PD-1 receptors on an exhausted T cell, it forces the T cell to stand down.
Right.
Checkpoint blockade drugs are essentially medications that rip those fake IDs away, allowing the immune system to recognize the tumor again.
But earlier you said terminally exhausted cells have broken brakes, like they usually don't respond to those drugs anymore.
Correct. But when the researchers combined the CRISPR deletion of these specific exhaustion genes with those anti-PD-1 checkpoint drugs in the mice, the tumor control was massively improved.
Wait, really?
Yeah. By deleting ZSCAN20 and JDP2, they took a T cell state that was completely resistant to our best current drugs and turned it into a state that responded beautifully.
So what does this all mean? I have to push back here for a second. We've proven the map works in a computer and we've proven it works in mice, but mice aren't humans.
Fair point.
I mean, we have cured cancer in mice 1,000 times over, only for those treatments to fail in human clinical trials. Does this same genetic rewiring actually apply to human T cells facing real-world exhaustion?
If we connect this to the bigger picture, the researchers anticipated that exact skepticism. The translational potential of any atlas hinges entirely on human validation. So they took human CD8 plus T cells and depleted ZSCAN20 and JDP2 in them.
And did the human cells respond the same way?
The human data mirrored the mouse data remarkably well. When those specific transcription factors were depleted, the human T cells showed a drastic reduction in the expression of those inhibitory receptors. The white flags came down.
But did they actually fight back? I mean, dropping the white flag is one thing, picking up a weapon is another.
They did. The edited human T cells showed a significantly boosted ability to produce potent effector cytokines.
Cytokines being the weapons, right?
Yes, these are the chemical flares and localized toxins that T cells secrete to actively destroy cancer cells. We're talking about a major, measurable increase in the production of interferon gamma, TNF, and IL-2.
So by identifying and firing these hidden, corrupt middle managers, they effectively hot-wired human immune cells to resist exhaustion and maintain their lethal edge against cancer.
That is the ultimate takeaway. This multi-omics atlas provides a foundational, highly precise blueprint. It shows us exactly how we can rationally design and engineer much more effective cellular immunotherapies like SCAR T cells, which currently suffer terribly from rapid exhaustion in solid tumors going forward.
This paper really is a massive leap forward. I mean, by decoupling the cellular program for exhaustion from the program for protection, scientists can now engineer T cells that are all fight and no fatigue. It's like giving our cellular workforce a limitless energy cheat code.
It really is.
But looking at this map leaves me with a final, somewhat provocative thought for you to ponder.
Oh, what is that?
If we can use this PageRank atlas to find the precise switches that prevent immune exhaustion in order to fight cancer, Could we theoretically use this exact same map in reverse? Imagine deliberately flipping these exact same switches, intentionally activating ZSCAN20 and JDP2 to intentionally induce exhaustion in overactive T cells. Could this be the blueprint for curing autoimmune diseases, where your body's immune system is relentlessly attacking its own healthy tissue? If we can fire the bad managers to cure cancer, maybe we can deliberately hire them to force a rogue immune system to finally take a rest.
Oh, wow. It's a fascinating mirror image of the problem. And honestly, that is exactly the kind of broader critical thinking that a foundational atlas like this enables. The map is drawn. Now it's simply a matter of how we choose to navigate it.
A question for another deep dive, perhaps. This paper is published under a Creative Commons Attribution 4.0 license, and full credit is provided in the episode description. Thank you for listening to One Paper Orbit-O, our research paper podcast. Our goal is to make peer-reviewed research accessible to everyone. If this episode was helpful, please share your thoughts and let us know which papers you'd like us to explore next. Thank you.